Let us have a discussion on ribonucleic acid, types and structure. Introduction. Ribonucleic acid, called RNA in short, is one of the nucleic acid essential for various biological roles in coding, decoding, regulation and expression of genes. RNA along with DNA, proteins and carbohydrates constitute the major macromolecules essential for all known forms of life. It is a polymeric molecule formed by different nucleotides. For a long time, molecular biologists have believed in central dogma of molecular biology, where DNA gives rise to RNA that gives to protein. Until recently, attention on RNA was given for its intermediary role in transcription and translation. However, that concept has now changed forever. The discovery that RNA has an important role in controlling gene expression through RNA interference (RNAi) has led scientists to recognize and reassess the significance of RNA. Now, with the popularization of RNA hypothesis, there is greater importance to the RNA. The phrase RNA World was first used by the Nobel laureate Walter Gilbert in 1986. The RNA World Hypothesis believes that self-replicating RNA molecules are precursors to all current life on Earth. According to this hypothesis, RNA is proposed to be the original form that stores genetic information. DNA is considered as a later addition. In today's lecture, let us discuss about the fundamentals of this important nucleic acid. Let us discuss about the structure and types of RNA and their biological roles. Structure. RNA is a polymeric molecule consisting of individual ribonucleotides. These nucleotides have a common structure composed of a phosphate group, a ribose sugar, which is a 5-carbon sugar molecule, and base, which is a nitrogen and the carbon containing ring structure. The bases are adenine, guanine, cytosine and uracil. The phosphate group are linked by a phosphoester bond to a pentose that in turn is linked base. The bases may be purines or pyrimidines. Adenine and the guanine are purines which contain a pair of fused rings. Cytosine and the uracil are pyrimidines which contain a single ring. The bases are often abbreviated A, C, G and U, the same single letter abbreviations that are used to represent entire nucleotide in nucleic acid polymers. In nucleotides, the first carbon atom of the ribose is attached to the nitrogen at position 9 of a purine or at position 1 of a pyrimidine. The acidic character of nucleotide is due to the phosphate group. This group under normal intracellular conditions release a hydrogen ion, leaving the phosphate negatively charged. A polynucleotide strand has a backbone consisting of repeating pentose phosphate units upon which the purine and the pyrimidine bases extend as side groups. The nucleic acid strand has an end-to-end chemical orientation. This polynucleotide chain can twist and fall into 3-dimensional conformations stabilized by non-covalent bonds. Base pairing in RNA is somewhat different from DNA. In the RNA, the bases may form hydrogen bonds between cytosine and the guanine, between adenine and the uracil, and also between guanine and the uracil. RNA is distinguished from DNA by the presence of a hydroxyl group at the 2' position of the ribose sugar. Presence of this functional group enable the molecule to adopt the A form geometry rather than B form commonly observed in DNA. The functional form of single-stranded RNA molecule usually requires a specific tertiary structure. The scaffold for this structure is provided by secondary structural elements that are hydrogen bonds. It leads to several secondary structures like hairpin loops, bulbs, and internal loops. As RNA is charged, metal ions such as magnesium are needed to stabilize secondary and tertiary structures. RNA can be double-stranded or single-stranded, linear or circular. RNA can also participate in a hybrid helix composed of one RNA strand and one DNA strand. In most cases, RNAs are single-stranded and exhibit a variety of conformations. Double-stranded RNA has two complementary strands and the found is genetic material of some viruses. Recently, there are reports of single-stranded covalently closed circular form of RNA. These circRNA are considered to arise through backsplice reaction. Differences in the sizes and the conformations of the various types of RNA enable them to carry out specific functions in the cell. Types of RNA. RNA may be genetic material or non-genetic RNA. RNA as genetic material is found only in plant, animal, and bacterial viruses. It is found in the absence of DNA. Genetic RNA has self-replication property. This type of RNA may be either single or double-stranded. In most of the cases, RNA are non-genetic. These RNAs are not self-replicating and always single-stranded. They depend upon DNA for multiplication. The process is a conventional one and known as DNA-dependent RNA replication. In most of the eukaryotes and the prokaryotes, there are three main classes of RNA involved in protein synthesis, namely messenger RNA, ribosomal RNA, and transfer RNA. Messenger RNA, abbreviated mRNA, carries information from DNA to the ribosomes, the site of protein synthesis in the cell. The sequence of the messenger RNA determines the amino acid sequence in the protein synthesized. Messenger RNA are complementary copies of genetic information taken from the nucleotide sequence of a gene in DNA. The messenger RNA constitutes only about 5% of the total RNA in the cell. Each gene transcribes one messenger RNA for synthesis of specific proteins. The messenger RNA contains the following regions: A. 5' cap region in most eukaryotes and animal viruses, which is formed by methylation of any 4 nucleotides. B. Untranslated regions are sections of the mRNA that are not translated. They occur before the start codon and after the termination codon. The first region is termed the 5' untranslated region and the 3' untranslated region respectively. C. The coding region that translates a long chain of protein after attaching with several ribosomes. This region is composed of codons that can be decoded and translated into proteins by the ribosome. It begins with initiation codon having the sequence AUG and responsible for starting protein synthesis. The termination codon is the last part of the coding region that signals termination of protein synthesis when it is complete. The sequences are UAA, UAG, or UGA. The coding region tends to be stabilized by internal base pairs that impedes degradation. This portion may serve as regulatory sequences in the pre-mRNA as exonic splicing enhancers or exonic splicing silencers. D. Poly-A tail is a long sequence of adenine nucleotide added to the 3' end of the pre-messenger RNA. This tail promotes export from the nucleus and translation and protects the messenger RNA from degradation. Ribosomal RNA. ribosomal RNA, abbreviated rRNA molecule, are important structural components in cellular ribosomes, essential for protein synthesis. It constitutes the major portion within the ribosomes. There are two subunits, the large subunit and small subunit. The large subunit ribosomal RNA acts as a ribozyme catalyzing peptide bond formation. This form of RNA is present in all cells. So, the genes that encode the ribosomal RNA are sequenced to identify an organism's taxonomic group and determination of related groups. Transfer RNA. Transfer RNA, abbreviated tRNA, is an adapter molecule that serves as the physical link between messenger RNA and amino acid sequence of proteins. Formerly, it was called soluble RNA. It is the smallest type of RNA. The transfer RNA can be described into its primary structure, its secondary structure, and its tertiary structure. The molecular structure of transfer RNA can be depicted by the cloverleaf model. The model revealed that the chain of transfer RNA consists of 2 ends and 3 arms. 2 of the arms have loops, D loop and T psi loop. 2 of the arms have loops, D loop and T psi C loop, with a ribosome recognition site. The D arm contains dihydrouridine while T arm contains pseudouridine. The third arm, called variable arm, has a stem with optional loop. One end of the chain serves as amino acid acceptor stem, usually attached to amino acid. The acceptor stem may contain non-Watson-Crick base pairs. There is a 5' terminal phosphate group. The CCA tail is present at the 3' end of the transfer RNA molecule. Amino acid is loaded into its position. The other end is called anticodon arm, containing anticodon that consists of 3 base sequence that pairs with a complementary base sequence in a messenger RNA. Bases that have modified by methylation may occur in several positions throughout the transfer RNA. Moreover, vowel pairing may occur between anticodon and the codon of messenger RNA. Base pairing of the transfer RNA anticodon with the messenger RNA codon proceeds from the 5' end of the codon. Once the first 2 positions are paired, exact base pairing of the third position is less critical. The third base of the anticodon can typically pair with either member of the purine or pyrimidine pair in the codon as appropriate. It means that it wobbles. It allows messenger RNA to be translated with fewer than 64 transfer RNAs that would be required without wobble. It is important because most organisms have fewer than 45 species of transfer RNA that can pair with 61 species of messenger RNA codons. Apart from these common RNAs, there are other types of RNA that are involved in post-transcriptional processes. Small interfering RNA. It degrades target messenger RNA. Once the messenger RNA is degraded, the messenger RNA cannot be translated into protein. This type of RNA exists as RNA-RNA hybrids and involved in RNA interference pathway. MicroRNA. It is responsible for translational regulation. They are generated by nucleolytic processing of the products of distinct genes. These RNAs are single-stranded and exert their regulatory action by binding messenger RNA and preventing their translation into proteins. Small nuclear RNA. It is used to process and chemically modified ribosomal RNA. These RNAs primarily guide chemical modification of other RNAs, but should not be confused with the guide RNAs that direct RNA editing in trypanosomes. Small nuclear RNA. It functions in a variety of nuclear processes. They are involved in process of splicing of primary transcript to form mature messenger RNA. Once the messenger RNA is degraded, it cannot be translated into protein. Both microRNAs and the small interfering RNAs represent new protein targets for therapeutic drug developments. The siRNAs are frequently used to decrease or knock down specific protein levels in laboratory experiments as a part of gene knockout technology. RNA interference. It is a biological process in which RNA molecules inhibit gene expression or translation by neutralizing targeted messenger RNA molecules. The RNA interference has great potential in suppression of desired genes. It involves microRNA and small interfering RNA. Now, RNA interference is a valuable research tool for biotechnology, medicine, and insecticides. The process reminds us of the importance of RNA in the evolution of life on Earth. It supplements the importance of RNA in support to the RNA World Hypothesis. Ribozymes. Apart from proteins, some of the RNA can also act as biological catalysts. Such RNA enzymes are called ribozymes. They exhibit many of the features of a classical enzyme. They have an active site, a binding site for a substrate, and a binding site for a cofactor. Ribozymes are usually associated with proteins that stabilize their ribozyme structure. Even then, it is the RNA that acts as a catalyst. Discovery of the ribozymes changed the view how life might have evolved. It supplemented the RNA World Hypothesis. The primitive form of life was believed to base entirely on RNA that can function as a genetic material and as the enzymatic machines. Conclusion. RNA molecules play an active role within cells by catalyzing biological reactions, controlling gene expression, sensing, and communicating responses to cellular signals. Ever since the proposal of the central dogma, role of RNA in protein synthesis is widely appreciated. Now, scientists discovered that RNA is also responsible for numerous other tasks like regulating gene expression and modification of different types of RNA. In prokaryotes, RNA are involved in virulence to regulation of bacterial growth. New forms and the uses of non-coding RNAs continue to be discovered. Clear understanding of these processes would help in understanding the secrets of life. Studies on RNA led to many important biological discoveries and numerous Nobel Prizes. However, much work remains to be completed to understand the secrets of RNA and its diverse nature. Thank you.